Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper. This is shift 1 that I will be discussing, and part C questions are being discussed. In this section I will be discussing the genetic questions, and the exam was conducted in March 2025. So 6 mutant yeast haploids H1 to 6 requiring histidine supplementation for viability were fused in pairwise combination to form diploids. Requirement of histidine was tested for the diploids. Okay. The results are shown below, where plus indicates diploid combination yielding histidine prototropes. So this is a complementation. Test, and wherever there is a plus sign, it shows that complementation has taken place. Wherever there is a minus sign, and one more point is that the if it is plus, means the mutation has taken place in two different genes. That is why they are complementing each other. Okay. Now, if it is a minus sign, it shows that there is no complementation, and the mutation has occurred on the same gene. That is the basis of complementation analysis. So first you see for the non-complementation groups, you group them. So histidine 1, this is 1, then histidine 3 and histidine 6. So they belong to the 1 cistron. Whereas 2, 4 and 5, they belong to different genes. 1, 3 and 6, they belong to the same gene. See, for histidine 2, if you see, okay, histidine 2, it will be the same gene. Then 4, okay, that also same gene, and 5 same gene. So 2, 4 and 5, they belong to the same gene, or the 1 cistron. Similarly, if you see histidine 3, it is the same thing that is repeating, that is 1, 3 and 6. We have already done 1, 3 and 6. We have grouped that. And in 4, if you see, it is 2, 4 and 5. We have grouped 2, 4 and 5 already. Then again it is 2, 4 and 5 and 1, 3 and 6. So totally there are 2 cistrons for histidine biosynthetic genes. So answer here is option 2. Then the table below shows type of chemical mutagen and the name of the mutagen. You have to match them. So here it is base analogs. Base analogs means they have structure similar to the naturally occurring DNA bases. And here the example is 5-bromouracil. Oh God, first itself is the answer, A4. The others are all wrong. A4 is, if you know base analog is 5-bromouracil, you can happily mark option 1 as your answer. Then intercalating agent means they insert themselves between the bases of the DNA, and it will interfere with the replication, repair, etc. So example is ethidium bromide B3. Deaminating agents means they remove amino groups from the bases of DNA, and it can lead to mutation. Example is nitrous acid C1. And alkylating agents means they donate alkyl groups. It can be your ethyl group or your methyl group. And example here is ethyl methane sulfonate. So D2, that makes option 1 as your answer. Then given below are few statements regarding gene actions observed in plants. So gene action means how these genes interact to produce a particular phenotype. And the gene action can be additive gene action or a non-additive gene action. Additive gene action, it is usually seen in the homozygous conditions. Here both the alleles contribute equally to the trait. That is in the case of additive gene action, and there is no dominance in additive gene action. Whereas in non-additive gene action, one allele will dominate over the other. or the expression of one allele of the gene is more stronger than the other allele, and it can show complete dominance as well as incomplete dominance. Then a non-additive gene interaction can also be epistatic also, where one gene masks or modify the expression of the other gene. That is non-additive gene interaction. Then coming to one more term is there, that is oligogenic qualitative traits. Now what is that? So oligogenic or qualitative traits means they are governed by one or a few genes, not by polygenic ones. Okay, difference are there. It is quantitative, whereas polygenic is quantitative, whereas oligogenic is qualitative. And oligogenic means they exhibit discontinuous variation, that is there are no intermediates there, whereas in polygenic inheritance you can see intermediates also. Then oligogenic traits, they are not influenced by environmental variation, whereas polygenic traits are influenced by environment also. And in oligogenic trait, the gene action, it is usually a non-additive type. It can be dominant or epistatic one. Then another term you are going to come across in the statements is genetic fixation. What is this genetic fixation? So it is a process by which one particular allele become fixed in the population. It can be either your dominant allele or a recessive allele. So what these breeders do is they will select for that good quality trait. Whether it is recessive or dominant, they will not see. If it is recessive, they will see that that is fixed in that recessive character become fixed in the population. That is, all individuals in the population will carry that specific allele. Where if it is the dominant allele that is of a good quality one for the plant, then they will go for the dominant allele to get fixed in the population. That is genetic fixation. Okay, now read the statements. In terms of pollination, self-pollinated species often exhibit additive gene action. I told you additive gene action is usually seen in homozygous condition. Self-pollination will result in the development of homozygous plants. So it is additive gene action. So answer is option A, A is correct. So that means 3 gone. Then non-additive gene action is less prevalent in cross-pollinated species. No, cross-pollination can result in gene interaction like dominance or epistasis like that. So it is non-additive gene action. Non-additive gene action, it will be more prevalent in the cross-pollinated species because you won't get homozygous conditions just like in self-pollinated species. So B is incorrect. So that means 1 also gone, 4 also gone. Happily you can go for 2 as your answer. Okay, then a simply inherited qualitative oligogenic trait. I told you what they are. Predominantly exhibit non-additive and epistatic gene action. That also I told when I was explaining. So C is also correct. Then a genetic fixation of superior genes will be more difficult with the dominance gene action. Yes. Now you know that dominance gene action is seen in homozygous dominant condition as well as in the heterozygous condition. Dominance gene will express itself and it will mask the expression of the recessive allele in heterozygous condition. So if you want to breed a trait that is controlled by recessive allele, in the presence of the dominant allele, it will be harder for the plant breeder to select for this particular recessive trait based on the phenotype of the plant. Why? Because in heterozygous conditions, the dominant allele will mask the expression of the recessive allele. So even if the recessive allele is present in the population, the plant phenotype will always be that of the dominant allele. So this will make fixation of superior genes more difficult with dominance gene action. So that is also correct. So A, C, D are correct. That makes option 2 as the answer. Then next one, in Drosophila melanogaster, A cross was performed and the resulting progeny are indicated. So females are mutant. So they are recessive. So yellow body color, sepia eye color, recessive character. Males are wild type. So that is dominant character, grey body color, red eye color. Then you got the F1 where females are grey body and red eye and males are yellow body and red eye. So the F1 progeny were sigmated. Okay, they were self-pollinated and selfed, and you got the F2 progeny. The following statements are made based on the above crosses and analysis of the progeny. So you have to identify which of the statement is true and which are false. Okay, so here if you see, you have got the females that are mutant and males which are wild type. These are the females and the males of the F1. Now I will tell you that you want to see which of these is X-linked and which is autosomal inheritance. Now if you see the F1 progeny, you can see that eye color is present in both male and female. There is no difference. Both male and female offsprings, they are having red eye color. Red eye color, why they got? Because red eye color is dominant. Male is wild type. Wild type male is having red eye color. It is dominant. So naturally red eye color is, or eye color is going to be autosomal inheritance. Whereas if you see the body color, you can see the female is grey and the male is yellow. After crossing, a female with yellow and male with grey. Now why does this happen? This can be because it is sex linked. Okay, we will see. Here is, I have scribbled here because by mistake I wrote the gene on Y. So I have scribbled here. I am extremely sorry for that. So yellow female means XX. It is recessive character. It is a mutant. So XGG is carrying the yellow color. It is homozygous and females are XX. And grey male means only one X chromosome will having the grey gene. The Y will not have any genes. So these are the gametes produced and these are the offsprings, and you find that wherever capital G is there, it is grey and it is a female. Here also it is grey and it is a female, and here also it is yellow and yellow. Why? Because the grey gene, that is the wild type grey gene, goes to the female from the male, because females inherit the one X chromosome from the male parent, and since the male parent is wild type, the female got the wild type body color, whereas the males offspring of the F1 generation, they will get one X chromosome from the mother and the Y chromosome from the father. They will not get X chromosome from the father. So naturally mother is mutant, so the males will be yellow. So that clearly shows it is a sex-linked inheritance. Okay, now see the true or false statements. The mutation leading to sepia eye color is located on an autosome. I already told eye color is autosomal inheritance. So the mutation leading to sepia eye color is located on an autosome is true. So 1 is true. That means 2 is not correct and 4 also not correct. Yellow body is a dominant phenotype. They already have given yellow body as mutant. So it cannot be a dominant phenotype. So B is false. So B is false means 3 also gone. That means 1 is your answer. Then 1/4 of the F2 progeny will be males and yellow body color. Okay, we will see that. F2 progeny is obtained by selfing F1 males and females. So F1 female is grey body color. Okay, here they have mentioned only for the body color. So we will see that one only, and we found that body color is X-linked. So females are grey body color, that means dominant XG small g, and the males will be yellow body color, means they will have X chromosome carrying small g. Now these are the gametes produced, and this is the female offspring. This is another female offspring which is yellow, and this is another female offspring which is grey. This is the male offspring which is grey, and this is the male offspring which is yellow. So what they have told, 1/4 of the F2 progeny will be males with yellow body color. Yes, out of all these F2 progenies, 1/4 will be males with yellow body color. It is correct. See here, male yellow body color out of 4 offsprings. So C is correct. Then in this dihybrid cross, as the F2 progeny does not show a 9:3:3:1 typical Mendelian ratio, the two genes can be assumed to be linked. How can it be linked? Because one character is present on the X chromosome, that is, it is X-linked, and another character is present on the autosome. So there is no chance they both will be linked. So the two genes can be assumed to be linked is not correct. So D is false. So true, false, true, false. That makes option 1 as the right answer. Okay, before coming to the next question, I will tell you what is this biparental mating and genome-wide association studies and how they are useful in QTL analysis. Now, what is this QTL? It is a region in the genome, and it is associated with a quantitative trait just like your height, weight, or disease susceptibility or crop yield. Before that, what is a locus? Locus is a specific location on a chromosome where you can find your gene or a genetic marker. And a QTL, it can involve multiple genes contributing to the trait. That is, a QTL can contain multiple loci contributing to the trait. Okay. QTL can involve multiple genes contributing to the trait, and it also means that it has multiple loci contributing to the trait. Okay. Now you have to see what is biparental mating. So in biparental mating, there is controlled cross between two parental lines. So from a large population of parents, you select the one with large, suppose it is for a large fruit size, you are going to select a large fruit size apple and small fruit size apple as your parents. Then you cross pollinate and obtain the F1. Then you self the F1 and you get the F2. Then you phenotype the F2, that is you measure the fruit size. Then you genotype the F2, that means you analyze the SNPs or the various other molecular markers like SNPs. Here it is SNP. And you identify the variants for SNP associated with the apple size. Okay, you identify the SNP which is associated with apple size. Then you do statistical analysis and you identify the genetic region, that is only a small region of the chromosome that is associated with the apple size. Then you identify the QTL that is linked to this apple size. Okay, that is what you do in biparental mating. So here only two parental lines are selected. Whereas when you do genome-wide analysis studies, you collect a large collection of apple accessions. That is, you collect different varieties of apple. It can be red, green, reddish, yellow, or pure red, or anything. You can collect all those varieties. You can collect different breeding lines of apple varieties. You can even collect wild varieties of apple varieties. Then you do the phenotyping studies, that you measure the fruit size of these accessions. Then there is a genotyping, that is you study the SNPs or you undergo next generation sequencing technologies to study these for genotyping the accessions. And then you scan the entire genome for the SNPs associated with the apple size. Then you determine the statistical significance between the SNP and the trait, that is your apple size. Then there is functional annotation, validation, follow-up. What does all that mean is functional annotation, validation, follow-up means you can predict the function of this particular gene using bioinformatics tools. Then you can analyze the expression of the gene in different tissues at different developmental stages, or you can even fine map the trait to identify the causal variant. Cloning studies also can be done here. So all these come under functional annotation, validation, follow-up. That is with GWAS and biparental mating. Okay, now next term that you have to know here is what is linkage equilibrium and linkage disequilibrium. So before coming to what is linkage disequilibrium, I will tell you what is linkage equilibrium. That is, it occurs when alleles at different loci are inherited independently. That means there is no non-random association between these traits. Okay, remember the term non-random association, because it will come in linkage disequilibrium, this term. Okay, that is if you have two genes on different chromosomes, that is if capital B and H occur in different chromosome, or if they are such that capital B is on and H are on the same chromosome but are far apart, that they can undergo recombination, then they are said to be in linkage equilibrium. So here if this one colors B, capital B is controlling the eye color, and this is for brown eyes and this is for blue eyes, and H is for black hair color and small h is for blonde hair color, you can get different combinations. That is brown eyes with black hair color, blue eyes with black hair color, brown eyes with blonde hair color, brown eyes with black hair color. So these are the various combinations that you can get, and you can say that this population is in linkage equilibrium. Now what is linkage disequilibrium? So here in linkage disequilibrium, the alleles at different loci. So this is the locus for hair color, eye color, and this is the locus for hair color, and they are inherited together. Okay, different loci, they are inherited together more frequently or less frequently than expected by chance. This is the term definition they have given for linkage disequilibrium. That is, they are inherited together more frequently or less frequently than expected by chance, and they show this non-random association. Okay, and this non-random association is because they are close to each other even though they are on different loci, and they are inherited together. That means there is less recombination taking place between them. That is linkage disequilibrium. Now coming to the question. So quantitative trait loci can be identified using two main approaches, biparental mating and genome-wide association studies. We saw that. Below are some descriptors related to these strategies. So these are the descriptors, and the table below present 4 incomplete statements regarding BMS and GWAS. Each statement can be completed using these descriptors. So which one of the following option correctly completes these statements? Okay. So first is, okay, we will see the option. The analysis is typically conducted on. So in biparental and GWAS. So first option they have given C. C is population derived from controlled crosses. Of course, you take only 2 parents to the maximum, 2 parental lines with different traits are selected. And GWAS you take very huge population. It can be random mating population. So C and D is correct here. Okay, so that means 2 gone and 4 gone. 2 gone, 4 gone. Okay, so that is it with the first option. Then the second one, the number of alleles that can be possibly be analyzed. So for biparental mating, it is limited to 2 alleles per locus. Yes. In biparental mating, I told you it is derived from 2 parental lines and each parent will contribute 1 allele. So the offsprings, they will get a maximum of 2 alleles per locus. Okay. So BMS, it is limited to 2 alleles per locus. So E is correct. Whereas in GWAS, it is multiple allele per locus because it usually uses a very large population when compared to biparental mating. And each locus can have multiple alleles, reflecting the genetic diversity of the population. That is, if I give you an example for color of the apple, biparental mating, you choose only 2 alleles, that is either red or green either of these, and the offspring can be having only red genotype, or it can have only green genotype, or it can have an intermediate one with red and green genotype. Whereas if you see the GWAS, you choose a diverse apple population and it can have multiple alleles per locus. That is, it can have the red ones, you can have the green ones, you can have the yellow skinned ones, and you can even have the pink ones. So these alleles can come in different combination in the offspring. It can be either red alone, or red and green, or red and yellow, or green alone, or red and pink, or green and pink, anything. So it is limited to 2 alleles per locus in case of biparental, and multiple allele per locus in case of GWAS. That makes option 3 as the wrong answer. Then the analysis relies on, so linkage based mapping. Yes, that is in the case of biparental mating. That is, you have, suppose there are 2 loci. Quantitative trait locus only you are studying. So you have 2 loci in that particular QTL, and it has that is A and B, suppose, and A is having 2 alleles, that is A1 and A2, and B is having B1 and B2 alleles. Then after crossing, you can get a combination of offsprings with A1 B2 or A2 B1. So then you analyze the recombination frequency, you estimate the genetic distance between the loci A and B. That is just purely your linkage based mapping. That is what is taking place in biparental mating. Then linkage disequilibrium mapping, that is seen in GWAS. That is, suppose it is studies the linkage disequilibrium between the genetic marker, and here I am telling you it is SNP that they are taking. It can be any genetic marker. Okay, here suppose it is SNP. Now there is SNP1 and SNP2 that are associated with the fruit trait, and if you find that when SNP1 is having polymorphism at A, that is adenine nucleotide, and SNP2 it is for thymine nucleotide. Now you find that whenever it is A allele or adenine allele for SNP1, it is always a thymine for SNP2. That means SNP1 and SNP2, they are in linkage disequilibrium. They are always inherited together. That is what you study in GWAS. And this presence of linkage disequilibrium between the SNP, it is useful in GWAS for identifying genetic variants that are associated with the trait. Then sample size needed to identify loci with small effect. Now what is this small effect? Small effect means as in polygenic trait. So many traits, multiple genes has it is polygenic trait. So multiple genes will have small effect on that particular trait. That is polygenic inheritance. So for detecting these small effect loci, it is difficult because it requires usually a larger population size and precise statistical analysis. Now both biparental mating and genome-wide association studies, they require a large population size. The only difference is that when compared to genome-wide association studies, the size of the population is comparatively less with biparental mating. But that doesn't mean biparental mating is having small sample size. It is also analyzing a QTL. It is involved in polygenic trait, and polygenic trait is for small effect loci. And for small effect loci studies, you need large population size. So both require large population size. So that makes option 1 as the right answer. Now next one. The pedigree, figure A, represents inheritance of a monogenic disorder. Monogenic means disorder means it is caused by, the disorder is caused by a single gene. Okay. A defective enzyme encoded by a mutant allele. Okay. Now there are capital A, small a, small a. So this is caused by a mutant allele. The functional and defective enzymes are resolved by a page. The allozyme pattern observed in some of the individuals in the family are represented in B. The frequency of mutant allele in the population is 0.04. Okay, now what is this allozyme? So allozyme are different versions of the same enzyme. Okay, different versions of the same enzymes. And how are they produced? By different alleles of the same gene. Now these are the different alleles for the gene height. So this is the tall gene, this is the dwarf gene. Now this will produce the tall protein and this will produce the dwarf protein. So they are different versions of the same gene, that is height gene, and they are produced by different alleles of the same gene. Got it? That is allozyme. Now how do you get these proteins resolved on the gel? Now suppose your offspring is having both the tall genes. So it will produce this protein. If it is the dwarf gene he or she has inherited, it will produce this protein. And if it is intermediate, it will produce both the proteins because both the alleles will produce proteins. Okay, T is producing protein and small t is also producing its protein. That is what you see in heterozygous condition. Now see the gel. So here you can see bands, 2 bands are there. So that means it is heterozygous condition. Okay. But here you see only 1 band and here also you see only 1 band. Whereas in others you see 2 bands. Now 3-1 you found that this is defective. 3-1 is defective. Even though the parents are not affected by the gene. Are not affected. Okay. You found that 3-1 is defective and it is producing only 1 band. So that means if lower band is present, then it is not, the enzyme produced by this allele is the one producing the lower band is more than sufficient to suppress the disease causing allele. Okay. So this is the functional enzyme. This is the functional enzyme and these are the non-functional enzymes. Okay. So this allele, the allele producing this functional enzyme is more than enough to suppress the expression of the non-functional enzyme. Okay, so that means it is haplosufficient. One allele is more than enough, okay, to suppress the non-functional enzyme. So that is haplosufficient. So that makes the statement A correct. The trait shows 100% penetrance. See here, 3-1 is defective and it is showing the phenotypic character also defective. Whereas 3-3, it is also producing the defective enzyme, but it is not showing the phenotypic trait. The phenotype is not expressed as defective. So that is not 100% penetrance. See, if it is complete penetrance means everyone with the gene, defective gene, will develop the disease. But here all the individuals carrying the defective gene are not showing the disease character or the disease phenotype. Only 50% are showing the disease phenotype, other 50% are not showing the disease phenotype. So it is not 100% penetrance. So it is 50% penetrance only. So that makes B wrong. So that means 3 gone, 4 also gone. Okay, the probability that a child born to individuals 2-3 and 2-4 will be homozygous. 2-3, 2-4. Okay, they are not having any disease. But they are, one is carrier, other we don't know what is the condition. Yeah, okay, that is also a carrier. They both are carriers of the gene. Will be homozygous for the gene is 1/4. You cannot say that with 100% guarantee, because it is, first of all, you told that the frequency in the question they have given frequency of the mutant gene is 4% and or 0.04, and there is 50% penetrance. So naturally you are not going to get it like the usual Mendelian answer is, that is 1/4. So C is not correct. Then both individuals 1-1 and 1-2 are necessarily heterozygous for the gene. That you cannot say. Okay. First of all, they have not given the gel data for 1-1 and 1-2. They have not given. They have not shown how many bands for the enzyme are there. So you cannot say they are heterozygous. See, this 1-1 can be capital A small a and 2 can be capital A capital A. Still then you will get offsprings which are not showing the disease, and also carriers also you can get. So you cannot say that they are heterozygous from the given data. So D is not correct. So that makes option 1 as the answer. True, false, false, false. So that's it. Thank you for watching my channel. Please do like, share and subscribe.